Now, inhibition of the host cell protein synthesis. Just now we studied inhibition of the host cell, the RNA synthesis. Now the protein synthesis, how the host cell inhibit the synthesis of the proteins. Most cellular proteins are assembled and synthesized in the ribosomes, which are called as polyribosomes, because after the translation, the ribosomes playing a very important role in the translation, and on the ribosomes, the proteins are translated. The nucleic acid genome is translated towards the protein in the form of the polypeptides. These are called the polyribosomes. The cellular protein synthesis is blocked by interference with synthesis and functions of the mRNA in some of the conditions. If the cellular mRNA synthesis is blocked, the rate of decrease of protein synthesis will also increase. Similar is the way if synthesis of the rate of the decrease of protein synthesis will increase, the mRNA synthesis may also be blocked. Here you can see the half-life of mRNA in actinomycin treated HeLa and L cells is approximately 3 hours. Similarly, we will take another example of the Newcastle disease virus later. The half-life of mRNA in Newcastle disease virus in the form and the HeLa cell lines, which are incapable, and which is, of course, the protein synthesis will be decreased. The cellular protein's inhibitory effect can be blocked by a few substances. And just like pyromycin, or we can take the example of the Newcastle disease in the form of the Newcastle disease virus and the HeLa cell lines, their protein synthesis is blocked with the pyromycin or parafluorophenylalanine. These are two substances which are capable of reducing the synthesis of the proteins, or which are capable of inhibiting the protein synthesis in the host. Newcastle disease virus is very important and is having high rate of mutation. Just like the avian influenza, they are causing the disease in the chickens, and they are first time were originated from the Newcastle in the USA. That's why this is called as Newcastle disease.